Welcome to NephJC, a Twitter-based nephrology journal club for physicians, students, and patients. This October, one of the works we will be discussing is a manuscript recently published by Su et al. In the September 7th issue of the journal Science. This paper presents the solved structure of the human PKD1, PKD2 complex with implications for research in polycystic kidney disease. Dr. Josh Waitzman, a nephrology fellow at Northwestern University's McGall Medical Center, will be giving us an overview of the paper in this video. Dr. Waitzman, for students and interns perhaps not familiar with autosomal dominant polycystic kidney disease, or ADPKD, can you tell us more about this disease?
Sure. ADPKD is a genetic disorder that causes many kidney cysts to form and enlarge throughout a patient's life. When these cysts grow, they squish normal surrounding kidney tissue and cause progressive kidney failure. About 1 in 1,000 people have a lifetime risk of developing ADPKD, and it is the fourth leading cause of kidney failure in the United States. Most cases of ADPKD are caused by mutations in two genes, called PKD1 and PKD2, which encode proteins of the same names. Current treatment strategies for ADPKD are really geared towards slowing the rate of decline in kidney function. But none of these approaches tackle ADPKD at the level of the genes and proteins that we know cause the disease. The PKD2 structure was solved in 2016, but only about 10% of patients with ADPKD have mutations in PKD2. The vast majority of patients with ADPKD, about 85% or so, have mutations in PKD1. And until this paper came out last month, the structure of PKD1 was unknown. Because this paper presents the structure of PKD1 and shows how it interfaces with PKD2, it provides important data in moving PKD research forward.
That makes sense. So for those of us who have not worked in the structural biology field, can you give us a broad overview of the methods used to solve the PKD1, PKD2 protein complex?
I'll do my best. Structural biology tools let us see the molecules and proteins that cause disease in 3D. Having a three-dimensional picture of a protein is often the key piece of information that allows for targeted drug discovery. Some structural biology tools, like X-ray crystallography and NMR spectroscopy, require a very large amount of purified protein in order to work. As a result, this has limited our ability to solve the structures of proteins that can't be purified in large quantities. A method called cryo-EM gives detailed structural information with much less protein. At first, it wasn't a widely accepted method, but it's become such a big deal in the last few years that its developers won the Nobel Prize in Chemistry in 2017. So here's how cryo-EM works. First, a protein dissolved in water is flash frozen in liquid ethane, which only remains liquid at super cold temperatures below -88 degrees Celsius. The superfast freezing process causes the water molecules around the protein to freeze in a way that they become invisible. This is the cryo part. Then, electron micrographs of the protein are taken to get detailed structural information. This is the same kind of electron microscopy that we use on kidney biopsy samples in a nephrology clinic. Finally, a computer helps process the images of the protein. Because the protein was caught floating in solution when it was frozen, some images will be of the top of the protein, others will be of the side of the protein, and some others will be of the bottom of the protein. And by classifying the images into different views, the computer helps us reconstruct a 3D image of the protein. Cryo-EM used to be called blobology because the resolution it could achieve was relatively low.
Wow, seriously. Blobology does not sound very sciencey.
Maybe not, but now because of advances in software and computing power, the resolution of cryo-EM is comparable to that achieved by X-ray crystallography, and structures obtained with cryo-EM can be used to guide drug development. So the authors of this paper used cryo-EM to determine the structure of a complex of the PKD1 and PKD2 proteins. Even though cryo-EM does not require as much protein as other methods of structure determination, it still took a lot of work to get enough protein to determine this structure. The authors estimate that about 50 liters, or 30 gallons, of cell culture was needed to get enough protein.
Wow, so that certainly is not trivial. After all that work, what did the authors find?
So in the structure that they determined, the authors found 1 copy of PKD1 and 3 copies of PKD2. the 4 proteins are arranged in a ring shape, with part of each protein copy in the cell membrane and part of each protein copy outside the cell membrane. They call the extracellular part a top domain. The PKD1 subunit has 2 additional parts, an N-terminal domain that resides inside the cell membrane, and a plat domain that sticks downward from the N-terminal domain into the cell. Honestly, when I looked at this picture the first time, it reminded me of a mullet, with a ring of proteins around the head, the thicker top domains on the top, and a tail made out of the N-terminal domain and plat domains that sticks out and down.
Yikes, I don't know what I think about a science structure looking like a mullet. What about a stingray?
Maybe if you squint kind of funny at it.
Or a sleeping otter, maybe?
I think it needs a rounder body. I guess we can think of it more like a raccoon hat like Davy Crockett would wear.
Okay, alright. So I can see that. What's the significance of this raccoon hat shape?
So the arrangement of the 4 proteins in this ring makes the complex look like an ion channel. But part of the PKD1 subunit, the part that should help ions travel through the channel, is actually positioned in a way that blocks ions from traveling through. Instead of leaving a nice pathway for ions to travel into or out of the cell, 3 positive charges from the PKD1 subunit stick out into the channel pore and block it. This may explain why no one's ever been able to show that PKD1 and PKD2 complexes act like ion channels, because maybe they're busted by design.
That's interesting. So how does this fit in with the prevailing hypothesis that defective calcium influx from the ER triggers signaling pathways that promote cyst formation?
It's difficult to say from this paper because there's not a lot of functional data. But it's hard to see how this kind of a channel could conduct calcium ions through it based on the structure that we have. Perhaps if it formed a different structure when bound to a small molecule, it's definitely going to take some further studies.
That's fair to say. So what is in the raccoon tail part of the complex?
So the tail has the N-terminal domain of PKD1 and the plat domain sticking off into the cell. These 2 parts look kind of like an upside-down ice cream cone to me, where the N-terminal domain is the cone and the plat domain is the scoop of ice cream headed for the ground. The authors can't find another structure that looks like this part of PKD1, so it's difficult to guess what it does. Other papers show that the plat domain helps with the localization of PKD1 to the right cellular location, but beyond that we don't really know what this N-terminal domain does for the protein.
So in a way, finding this part of the structure leaves us with more questions than answers.
Absolutely. But the authors do show us that the top domains of each subunit interact with each other, and what they present here is novel. In a previous structure of a PKD2 only complex, each top domain interacted with its 2 neighboring top domains. I've depicted this here with megablocks that I borrowed from my son. In contrast, in the PKD1, PKD2 structure, the PKD1 subunit only interacts with one of the neighboring PKD2 subunits, leaving a gap in the side of the protein. The authors think that this is why there's a 1-to-3 complex. A 2-to-2 complex, which I've kind of made up over here, wouldn't have interactions between the 2 halves, and the 2 halves could just drift apart.
Ah, that makes more sense. Where does this leave us in terms of understanding ADPKD in general?
So having a structure of the PKD1, PKD2 complex is a really important step toward understanding the molecular processes that cause disease. In Figure 7 of the paper, the authors map all of the known mutations in PKD1 and PKD2 onto their structure.
So let's say I got my genome sequenced and I know I have a coding mutation at the PKD1 locus. How do the findings from this paper bring me closer to a more definitive treatment plan?
So structure tells us a lot, but it doesn't tell us everything about function. Since we still have open questions about what this complex does, we don't yet know how individual mutations will affect the activity of PKD1 and PKD2. Good structure papers cause us to ask more questions than they answer, and that is certainly true here. We still don't know what the PKD1, PKD2 complex does when it's working. Is it always a broken ion channel, or is there some small molecule that opens it as part of its normal function? The mutations that prevent normal assembly of this complex, could we develop antibodies or other drugs that pull together working complexes? In diseases caused by PKD1 mutations, do PKD2 only channels form? And could those channels be targeted by drugs to restore health?
Well, those are definitely some important questions that will require more research to answer. Thank you, Dr. Waitzman, for breaking down this very important paper for us. Join us for the NephJC discussions on October 9th and 10th, where we can dive into these concepts further. We look forward to tweeting you there.